So CRISPR just released a video about how during the 1950s, scientists released millions of sterile, radioactive, flesh-eating flies into Central America in order to cause their population crash. Note, it wasn't the radioactivity that caused them to become flesh-eating. They were already flesh-eating, although that would have been pretty cool. Now, the reason why this works is that this species only mates once before they lay their eggs, and they only are actual flyers for 5 days. So you can rapidly cause a population to crash by flooding the population with millions of sterile males. Now the problem is that you have to keep on doing this again and again in order to essentially top up the number of sterile males within that population. And so this is not the optimal way to achieve genocide. So I think we can do better. And that's where gene drives come in. Hi, I'm Angus. I have a background in evolutionary biology and genetics and education, and today we're going to be talking about how to destroy a species. So let's say you're super into genocide and killing all the bugs in Helldivers 2 just isn't cutting it anymore. Get some! Get some! Get some! This is the video for you. So genetic modification can be a little bit of a controversial subject, but let's say that you're a mad scientist and you really want to find the best way to completely eradicate a species. How do you do that? You might think, hey, okay, I'm just going to edit this gene and I'm going to cause it to make all males sterile, or something along those lines. Now, the first problem that you're going to get is Mendelian inheritance. So Gregor Mendel was an Augustinian friar and abbot who seemed to have a lot of time on his hands, being born before social media and video games, and so became a pretty prominent mathematician, botanist, astronomer, and most importantly, geneticist. Well, technically genetics wasn't really a thing back then, they didn't know what it was, but he became known as the father of genetics. And this is actually pretty interesting because he studied pea plants, and by breeding various pea plants together, he managed to infer the existence of heritable factors and completely got it. He managed to figure out, recessive alleles and dominant alleles. They weren't called alleles back then, but he figured out how inheritance works. In essence, when you're creating gametes, which are the individual sex cells, like sperm and eggs, the way that your cells divide, known as meiosis, causes your pairs of chromosomes to split into single chromosomes. And therefore, when a new individual is made, it receives one chromosome from its mother and one chromosome from its father. So what that means is that when you genetically modify an organism and that gene can be passed down, there's still only going to be a 50% chance that the next generation is going to have that specific modified gene. This is really inefficient. And nature has found a way.
Life, finds a way.
To basically say, fuck Mendel and his stupid Punnett squares. Genes are inherently selfish. They want themselves to be passed down. They don't care about the organism as a whole. In the same way that individuals of a population don't care about the population as a whole. It's about passing down the individual genes. And within an organism, it's about passing on each single gene. Each gene is essentially out for itself. Okay, that's a radical oversimplification, but stick with me. Evolution is essentially a multi-billion-year experiment. We add a couple of things over here, we mutate a nucleotide, throw things at the wall, see what sticks, and go from there. Again and again and again and again. Just iterating until eventually you find something that increases the survival of an organism, increases the fitness of an organism. Consequently, natural gene drives have evolved. So for example, during meiosis, so during the process in which a cell turns into an egg, there are 2 stages of division. So one cell turns into 4 egg cells. During each of those divisions, one of the 2 cells becomes what's known as a basal body, which is basically a shriveled up prune so that the other cell can have all of the juices, all the good stuff, all the mitochondria and the organelles and the cytoplasm. So some genes have found ways of increasing the chances that they are actually going to be on the cell that becomes an egg rather than the one that becomes a basal body. This is known as meiotic drive. Meiotic drive. Also, you get what are called selfish genetic elements, which I love. So, for example, transposons. And transposons are sections of DNA that are able to transpose themselves, move from one chromosome to another, or move around on a chromosome, basically skipping to the head of the queue. So, continuing humanity's long tradition of stealing from nature, see airplanes, camouflage, and looking absolutely fabulous, we've essentially stolen this idea from nature and also co-opted an existing process in order to allow us to do it, namely CRISPR-Cas9. CRISPR deserves a whole video of its own, and let me know in the comments if you want to see one. In essence, CRISPR involves taking a stretch of DNA that you want to be inserted into a genome, as well as adding a certain enzyme that's able to then identify the section of DNA that you want to paste that into, and then cut the DNA open, put it in, and close it. The process behind CRISPR comes from natural defense mechanisms within bacteria. Bacteria are in a constant war with viruses, and as a way to protect themselves against viruses, they essentially cut out sections of viral DNA and paste them into their own genome in order to be able to identify them more easily later on down the line. Now, what's clever about this is that it's really, really specific and allows for sections of DNA to be added in very distinct places, very effectively and very cleanly. This process has been one of the biggest breakthroughs in biology since the discovery of DNA. So how this has been used for gene drives is a little bit different. Not only are you adding in the section of DNA that you want, but you're also adding in the instructions in order to facilitate the CRISPR process to happen later on. What this means is that when the chromosome gets passed down from, let's say, the father, and it lines up with the chromosome from the mother, it's able to cut open that chromosome and then copy in that section of DNA, thereby bypassing Mendelian inheritance entirely and ensuring that every copy of that chromosome from that point on down the generations will have that gene that you want to be in there. Now, this is where things become a little bit interesting, because that gene can do anything that you want it to do. And for the purposes of genocide, it can make the organism sterile. So we have the mechanism to make sure the genes get passed down. What do we do with that? Well, there are a couple of mechanisms that would ensure that a population gets completely wiped out. The first one, as I like to call it, is creating mosquito incels. And I'm using mosquitoes in this example because, like, fuck mosquitoes, like, that's not a hot take. Basically, you can make a gene that shreds the X chromosome, meaning that every time a Y chromosome lines up with an X chromosome, it destroys the X chromosome. A second way that you can do it is to make all females sterile, but have that gene be passed down on the Y chromosome. Therefore, every single daughter will become sterile, and males can still reproduce. So the male mosquitoes are still going around creating more male mosquitoes, but all the daughters will be sterile. A couple generations later, no more mosquitoes. And alternatively, let's say you're trying to be like somewhat ethical about it, you can breed mosquitoes that are resistant to Plasmodium, which is actually the parasite that causes malaria. Mosquitoes don't cause malaria. Plasmodium, the parasite that they have inside of them, that they squirt into us when they bite us, that's the thing that causes malaria. We can make mosquitoes resistant to that, basically eradicating malaria altogether while keeping mosquitoes available as a food source in the food web, somewhat less problematic. Right, so gene drives, super powerful technique. You might be asking, hey Angus, if this is so amazing, why haven't we used this yet? Well, this is where the problematic stuff comes in. It might sound wonderful to completely eradicate all mosquitoes, but you don't know what the downstream effects of that are going to be. Humans are really bad at predicting the consequences of our own actions, and you don't know like exactly how they fit into this domino effect of interactions and interrelationships. You might end up completely destroying an ecosystem. For example, you could potentially use this in order to eradicate an invasive species. There have been lots of islands in which mice and cats have been introduced that have messed up the natural ecosystem. And so you could genetically modify some of those mice with a gene drive to destroy that population. But then all you need is one of those mice to board a ship and head back to the mainland. And then all of a sudden, all mice dead, all gone, mice dead, and probably everything else dead because of that.
Now, everyone is dead.
And furthermore, it's not like trying to eradicate an entire population based on their DNA has ever been misused or gone badly at all. So, somewhat problematic. So there you go, gene drives. Now you know something that you didn't know before, maybe. If you liked the video, comment, share it, subscribe. Tell me how I got everything wrong in the comments. Leave me some feedback. I base all of my sense of self-worth on external validation. So yeah, I need this. And also I don't know how to end YouTube videos yet. So I'm just gonna do this.